A very good afternoon, ladies and gentlemen. We expect to begin the program in about 5 minutes from now. You may please take your seats. We request and appreciate if you could kindly put your mobile phones into silent mode. Thank you very much. Very good afternoon, ladies and gentlemen. All of you, please be seated. May I now request the dignitaries to pay floral tributes to the portrait of Dr. Nayudamma and occupy their seats on the dais, please. A very warm welcome to all the delegates from the industry, academia, and the institute for the Nayudamma Lecture 2023. We begin today's function with the invocation. May I request you all to please rise for the invocation. May I now request Dr. K.J. Sriram, Director CSIR-CLRI, to welcome the gathering, please.
Good afternoon, Professor Majumder, my friends from the industry, my colleagues from the institute and other neighboring institutions, the students of this institute, right from the PhD programs to the UG programs. A very good afternoon to one and all of you. This lecture, under the title of the Nayudamma lecture, is in honor of the second director of this institute, Dr. Y. Nayudamma, whose 100 years was celebrated with lot of fanfare by this institute just a few months back. Dr. Nayudamma was, though he was the second director of this institute, was already being put for training to be the director, and therefore we had in Professor B.M. Das, the first director. What Dr. Nayudamma brought to this institute through a Tanners get-together which was an industry institute interaction to decide on what research this institute does on the leather technology front, is all known to us. But what Dr. Nayudamma did also was that he did not promote a technology per se, but he ensured that the science associated with this protein called collagen was also brought into this institute with great honor. He therefore set up, in the growth of this institutional path, laboratories for chemical sciences, laboratories for biological sciences, and so on and so forth. And also worked very closely with Professor G. and Ramachandran to look into the aspects of collagen. He brought in experts from across the world to discuss about collagen-based biomaterials, on how we could use the other parts of the animal other than the meat and the skin and add value to the cottage industries. But in all these things, one aspect which he ensured was that this laboratory also imbibed the science component of those things. And for this, he also, if I go back and look at it, he possibly was the director who has brought in the maximum number of Nobel laureates into this institute. He had a tremendous connect with the global scientific fraternity, and this institute had been opened up to learn from others. Today, by having Professor Majumder with us, we are in a position to understand more about genomics, and thus, through that understanding, look forward to what can we do in our sciences as well. As a part of this 1 week, 1 lab, which is going on now, today morning I was told by my friends, the volunteering groups, that the students of computer science were asking what can they do in leather field or what they can do in the footwear science. There were students from the BSc, MSc backgrounds also asking several questions. In all these things, we ultimately realize that leather science or technology is not working in silos, but having to imbibe the science and the essence of technology from every possible field that we can see on Earth. Therefore, I'm very sure this lecture in honor of Professor Nayudamma that we are going to have today will open up our minds to better understanding of greater things to come. In all these things, I would also request my students of the BTech program to really look into what more can we learn beyond the leather science that we are all studying in our curriculum. With this, I welcome every one of you to this program. My special words of thanks to Professor Majumder, because I had initially spoken to him for the this program to be done in Kolkata, but we had to change it. And he could somehow find time to be with us today in between his connectivity travels to find time to be with us today. I'm extremely thankful to you, sir, for this. And your presence in CLRI would add wonders to our organization. Thank you very much.
Thank you, sir. Now I request our director to give a traditional welcome to our speaker today, Professor Partha P. Majumder. Thank you, sir. In memory of Dr. Nayudamma's contributions to science and technology, Y Nayudamma Lecture is being organized every year, and this year it is also part of 1 week, 1 lab celebrations of CSIR-CLRI. Let me read out the bio brief about the speaker today. Professor Partha P. Majumder, National Science Chair, Scientific Excellence, SERB, Government of India. Distinguished Professor John C. Martin, Center for Liver Research and Innovation, and Emeritus Professor, Indian Statistical Institute, India, has made significant contributions to human, statistical and population genetics and genomics. He has developed methodologies for mapping human disease genes, identified genomic factors underlying many diseases, notably oral cancer, and has reconstructed the ancestries and relationships of ethnic population groups of India and Asia using genomic methods and tools. He has immensely contributed to capacity building in human and statistical genetics in India. He has over 350 scientific publications in journals of international renown. He has founded the National Institute of Biomedical Genomics, India. He is an elected fellow of all the three science academies of India, of the West Bengal Academy of Science and Technology, the World Academy of Sciences, and the International Statistical Institute. He has also served as the president of the Indian Academy of Sciences and the West Bengal Academy of Science and Technology. He is a member of the executive committees of the International Human Cell Atlas Consortium and the International Common Disease Alliance. He is the Indian National Coordinator of the International Cancer Genome Consortium. He has provided service to the UNESCO and the WHO. Now, to read out his awards, he has been awarded the G.N. Ramachandran Gold Medal 2021 by the CSIR, Barclay Memorial Award 2020 by the Asiatic Society, Sir P.C. Ray Memorial Medal 2020 by the University of Calcutta, Golden Jubilee Commemoration Medal 2018 by the Indian National Science Academy, Centenary Medal of Excellence 2014 by the School of Tropical Medicine, Twas Prize in Biology 2009 of the World Academy of Sciences, G.D. Birla Award for Scientific Research 2002 by the K.K. Birla Foundation, New Delhi and Kolkata, the Om Prakash Bazan Award in Biotechnology 2001 by the Om Prakash Bazan Foundation, New Delhi, the Ranbaxy Research Award in Applied Medical Sciences 2000 by the Ranbaxy Science Foundation, New Delhi, and the New Millennium Science Medal 2000 by the Indian Science Congress Association and the Council for Scientific and Industrial Research, the Government of India. Now, I take the pleasure of inviting Professor Partha P. Majumder to deliver the Nayudamma Lecture 2023 on Genomic Medicine, What's All This Excitement? Please, sir.
Good afternoon, everyone. Thank you very much, Professor Sriram, for asking me to deliver this talk. Can I get my slides? I want to start this talk with three. Okay, all right. Put it in the pocket. So like I said, that I'm hugely honored to be called upon to deliver this talk. I have looked up who the previous speakers were on this particular series, the Nayudamma series of lectures, and they were very stellar people, and I don't actually belong to that league of people who actually delivered the previous talks. The second point that I want to make is that I actually did meet Dr. Nayudamma, and I was a student at that time. In 1972, the Indian Science Congress was held in Calcutta, and Dr. Nayudamma was there. I can't recall anything of what I had heard. I did sit through his talk. What I do remember is that he was a very tall and charming gentleman. That's all I remember of Dr. Nayudamma. And my association with leather is essentially with the human skin, and I've done some work on human microbiome that affects the human skin. And that's all is my association with leather. But this morning I had a fantastic discussion with a group here and learned a lot about the kind of work that goes on, and it was a kind of a revelation because I didn't know many of the things that relates to leather and leather research. So it's been a fantastic, my travel to this institute has been wonderful. I've only spent a few hours, but I've learned a lot. I really don't think that this particular topic is of great interest to the institute in a professional way. But most of us today are interested in genomics, especially after the pandemic. Everybody talks about genome sequencing. So whatever it means to different people, everybody knows the phrase genome sequencing. I'm not actually going to dwell on the technologies of genomics, but essentially going to tell you that there is a huge amount of excitement, not because of this phrase that people know, but generally there's a lot of excitement about genomics. And genomics as applied to medicine, as applied to human health and disease. And I'm going to start with a few examples and tell you how medicine is becoming more and more precise in terms of its treatment, in terms of its management, by understanding the genomic backgrounds of human health and disease. That's essentially what I've set out to do in today's talk, and I think it will be of general interest. But again, there will be certain technical portions in this talk, and I will try my best to explain those technical portions in as lucidly as I possibly can. All of this, we talk about precision medicine, we talk about genomic medicine, essentially because there's a lot of heterogeneity in human health and disease. Not every one of us is vulnerable to the same kinds of diseases. Not every one of us has similar features of health. And so there's a lot of heterogeneity that there is in human health and disease. And I will start with two sand dunes. These two sand dunes look exactly the same, and but they are essentially quite different in the sense that if you scratch the surface of these sand dunes, what you find is that even though they are similar looking, the grains of sand in the first sand dune are much more fine compared to the second sand dune. The grains of sand in the second sand dune, which is much more coarse. So even though you might have, let's say, tumors in the body, cancers in the body looking exactly the same, but if you look at it, if you take a slice of the tumor and look at it under the microscope, you would find that there are huge differences. These differences are important to understand. These differences are important in terms of their treatment. So you need to understand why these differences have arisen. And once you understand those, the hope is that you can manage the disease better and you can probably treat those patients in a better way. And then medicine becomes much more precise and genomically driven. So like I said, similar looking tumors may not be homogeneous. Exactly what I said, that if you look at this particular cross section of the tumor under a microscope and this cross section of the tumor under a microscope, as you can already see that the cellular composition, the tissue composition of these two tumors are grossly dissimilar. What we're going to talk about is genomics. So I really don't need to define what genomics is, but I'll quickly go through what exactly we mean by genomics, because not all of you may be exposed to this particular word. All of the hereditary information that's contained in the DNA of each cell, each one of our cells contains genomic information, and that's the hereditary information, the pool of the hereditary information that's contained in the DNA of each cell is called the genome. And the study of the genome is genomics, and that's a discipline that we seek to study and understand the entirety of hereditary information contained in an organism. There are, in the human, different organisms would have different amounts of DNA, different lengths of DNA. In the human, there are 3 billion alphabets in the human genome, and not all alphabets look exactly the same. The sequence of alphabets don't exactly look the same in different individuals. There is variation among the genomes of different individuals, and these variations actually provide us with preponderance to certain or vulnerability to certain kinds of bacteria, infectious diseases, vulnerability to certain other kinds of cardiovascular diseases, and so on and so forth. So we need to understand all of this in order to understand our own health and vulnerabilities to different diseases. We talk about precision medicine. Precision medicine is, of course, genomically driven. And essentially what it means is that we want to use genomic information that's contained in the cells about a person's disease to diagnose or treat the disease. So that's essentially precision medicine. So we want to know not just the clinical features of the disease, but also to understand what are the underlying genomic features pertaining to that disease in that particular person in order to be able to diagnose or treat the disease. So in this particular sense, when we want to talk about genomic information, unless we talk about genomic information at the level of a single individual, this kind of heterogeneity that one sees, observes, this heterogeneity hurts, because if you didn't understand this heterogeneity, you're going to treat all tumors, all patients with the same kind of treatment or manage them with the same protocol, and that's not going to work for all patients because of the heterogeneity. So we need to understand this heterogeneity and use this heterogeneity for the purpose of treatment or for the purpose of disease management. I'm going to talk a lot about cancers, and I want to talk about two classes of genes that are involved in cancer. One class of genes is called tumor suppressors. So these tumor suppressors will produce proteins normally, and those proteins will keep tumors in check, will not allow tumors to grow. So these are normal growth inhibiting tumors, and as you know that most of these cancers will be uncontrolled cellular growth is a hallmark of these kinds of cancers, and tumor suppressors will keep this uncontrolled cellular growth in check. The other class of tumors, so when there are mutations, when there are changes in the normal tumor suppressor gene, the protein that it produces, each gene produces a protein, the protein that it produces becomes defective and sometimes non-functioning, as a result of which the cellular growth that was kept under control will go haywire, and then cellular division will actually become uncontrolled and a tumor will appear. So this is one class of genes that are normally expressed, all the time. But if there is a mutation in the gene, if there is a change in the gene, then it might not express itself in the normal way, and therefore its normal function will be obliterated, and obliteration of the normal function of a tumor suppressor will mean uncontrolled cellular growth and therefore development of a tumor. The other class of genes that are involved in cancer, they're called oncogenes. These genes are like sleeping giants. They're just genes that are there in the genome, but they're not expressing. Normally they don't express. And when there is a change in the gene, when there is a mutation of the gene, sometimes these mutations will wake up this gene, which essentially means that they will start producing proteins. And these proteins are terrible proteins, are harmful proteins that actually will cause the cancer. So it will accelerate cellular growth, and it will cause cancers. And therefore, both of these classes of proteins are important. Both of these classes of genes are important. The point to remember is that tumor suppressors are expressing themselves all the time, normally, and a mutation is shutting them off. The oncogenes are sleeping all the time, and the mutation is actually waking them up. And once a gene is shut off, the tumor suppressor gene is shut off, it fails to regulate cellular growth, and once an oncogene is activated, then it leads to uncontrolled cellular growth. In both cases, tumors happen or cancers happen. Only about 10% of any cancer is due to inheritance of a faulty gene. So as we know that we inherit genes from our parents, and we are born with those genes at the time we are born. So if there are changes in the parental genes, if there are mutations in the parental genes, then we are inheriting the mutant genes from the parents, and those mutant genes can be faulty. But in cancers, what one has seen is that only about 10% of cancer is due to inheritance of a faulty gene. What happens to the remaining 90%? They are all because of mutations in either a class of tumor suppressors or in a class of oncogenes. What happens to them? They haven't inherited those from their parents. Then what happens? How do they get cancer? So the cancer we now know is that those are not inherited, but they happen during the life course of this particular individual who has gotten the cancer. So essentially, lots of changes take place in our own genomes all the time. Most of them we are not even aware of, but ultimately certain mutations that are happening in our lifetime, they are happening in the body cells, they are not happening in the germline, meaning that those are not the cells that we will pass on to our offspring. So they happen in our body, they happen in our tissues, and they die with us. They are not transmitted from us to our offspring. So those are somatic mutations. So most cancers are due to somatic mutations. We haven't had the technologies to be able to study somatic mutations up until 20 years ago. Just technologies did not exist for us to be able to study somatic mutations in any great detail, in any accelerated or high throughput manner. It's about 20 years ago that technologies became available, and all of us started to ask the same or similar question: what is it that's driving a specific form of cancer? And we have looked at oral cancer, because oral cancer is a very prevalent form of cancer among men, especially among men in India, primarily because of the tobacco chewing habit. And we've looked at, I've spent last 10 or 12 years of my life just researching on oral cancer and trying to understand what causes oral cancer, what kind of somatic mutations happen, what are the genes that drive normal tissue to becoming cancerous tissue in the oral cavity. But I'll come to that at the end. What I want to do is motivate you, try to motivate you to understand what genomic medicine is all about. So I'll start with work of others. I'll start with work that's something that you can relate to, some fantastic figures and stories. So if you look at breast cancer, worldwide about 1.6 million new cases of breast cancer are diagnosed every year, leading to over 500,000 deaths. So those are startling figures. You have 1.6 million new cases. Breast cancer, of course, is in women mostly. It also affects men, but very rarely. It mostly affects women. And over 500,000 deaths happen every year. 40 years ago, if a woman was diagnosed with breast cancer, the chance of survival was only about 50%. Today, if a woman is diagnosed with breast cancer, chances are that the woman will survive for a long period of time, and the chance is about 80%. So this is a very significant difference from within 40 years. The change from 50% to 80% is significant. Now, how did that change come about? The way that change, this kind of change came about is that in about 20% of breast cancer, the cells make too much of a protein known as HER2. This is only in about 20% of breast cancers. Breast cancers are also very heterogeneous. Like all cancers are, breast cancers are heterogeneous. In about 20% of breast cancers, the cells make too much of a protein known as HER2. So we will concentrate on this one example. This example is good because any woman who has where HER2 mutation, where HER2 protein is being made in large quantities, and cancer has set in, these cancers tend to be aggressive and fast growing. So these cancers, if we understand these cancers, we probably will be able to manage these cancers better. So we have now understood what happens in this particular type of cancer. This is chromosome 17, and there is HER2. This is the long arm of chromosome 17. This is HER2. This is exactly where this particular protein known as HER2 is produced. And this understanding what leads to this overproduction of this protein and what this overproduction does to the health of this woman has provided us with lots of answers. Usually there are two copies, but up to four copies of this particular gene, HER2. HER2 comes in multiple copies in an individual. Usually, like I said, they come in 2 copies in an individual, but up to 4 copies may be okay. But once it becomes more than 4 copies, then it's producing too much of this protein. And this particular protein is known as human epidermal growth factor receptor protein. And this protein is expressed at the cell's outer surface. What it does is it receives signals from the surrounding environment that trigger the cell to grow and survive. So like I said, that the hallmark of cancer is unregulated, uncontrolled cellular growth. So once it receives signal for the cell to grow and survive, because if it survives for long, then the tumor remains for a long period of time. And when you have many copies of HER2, there is more protein production. And then there is an overload of receipt of signals and uncontrolled cellular growth. So essentially, what one has done is to understand that there are multiple copies of HER2. Multiple copies mean more production, and then one scientists have done a lot of functional work to figure out what happens if there is more protein production. What it normally does is to receive signals, keep signals within control, and keep the cellular growth under control. But when there is a lot of overproduction of that particular protein, then tumors happen because of uncontrolled cellular growth. If you look at survival, the individuals in whom there is no amplification of this gene, that's no amplification. This is where it is amplified. And if you look, amplified meaning multiple copies and therefore multiple amount of greater amount of production of the protein. So if you this is called a survival curve. It doesn't matter what, if you look, cannot read it, doesn't really matter. I will just tell you what the conclusion from this graph is. If you do not have an extra HER2, your overall survival probability is greater. So this is this curve is above this particular curve where there are more than 5 additional copies of HER2. This is time in months after it's detected, and so what one is tracking is that for every month, how long does the patient survive? And if you don't have extra copies of HER2, you survive longer than if you had extra copies of HER2. So the take-home message is that extra copies of this particular gene called HER2 hurts. Once that was understood, once the function was understood and it was related to the survival of the woman who has breast cancer, one was able to, so essentially it's overproduction. So if you can somehow retard the production of that particular protein, you're in business. So it's a drug called Herceptin was actually identified that would block this particular protein and dramatically block this particular gene, and therefore reduce the amount of protein that's produced by the gene or the additional copies of the gene, and that dramatically reduces the risk of cancer coming back. So Herceptin also has much fewer side effects. Oops. I must have pressed the wrong button. Can you go back to the first slide, please? Go back. No, go to the first slide. Yeah, that's fine. That's fine. I'll do it now. So it also has much fewer immediate side effects. For example, as you know that if you undergo chemotherapy, then there is you lose taste, you lose your hair cells, and so on and so forth. I'm not going to get into the technical details of this, but essentially, Herceptin was designed to attach to this particular protein, and that would block the effect of that protein, as a result of which there would be dramatic reduction in risk of cancer coming back. What is happening? I'm just pressing this. I'm just pressing this one. That's the forward button, right? Yeah, okay, all right. No, that's not good. No, you can't operate from there because I'll lose my. I'll do it. No, there's no video. All right, so it's okay. So essentially, what genomics understanding of genomics has done is to be able to save lives of women by tailoring drugs more precisely using information on their genome. So now we know that HER2 essentially overexpresses, and we now have understood the structure of HER2, blocked HER2, and therefore we are able to save lives of women. So this is again drugs that are tailored more precisely by using information on the genomes of individuals who have breast cancer. So that was women. We now turn to children. And if you look at children, so these are children, this is the percent of children with certain form of leukemia, a specific form of leukemia, doesn't really matter what that specific form is, but it's leukemia, and children are affected with leukemia. And if you look at the percent surviving, the number of the percent of children who were afflicted with leukemia, this form of leukemia, in 1960, only 4 of 100 children survived. Today, in 2014, 92 of 100 children survived. And in 2021, close to 99 of the 100 children who are diagnosed with this form of leukemia survived. How did that happen? So essentially, again, understanding of cells of these individuals, cells of these unfortunate children, and doing a lot of molecular biology work has made us understand how this happens, and as a result of which, we are able to get better drugs and be able to control. So, normal chromosome 9, there is a gene called ABL, and in normal chromosome 22, there is a gene called BCR. Sometimes the chromosomes, during their process of duplication, etc., some wrong things happen, and one of the wrong things that happens oftentimes is translocation. So sometimes there's a breakage of chromosomes, and this particular part of chromosome 19 breaks and gets itself attached to chromosome number 22, which has this BCR. And so these two come together, the BCR and the ABL come together, and they start producing independently. They produce 2 different kinds of proteins. And when they come together as a result of translocation, they produce a fusion protein called BCR-ABL fusion protein. And this particular fused chromosome is called the Philadelphia chromosome. Again, there's now a book called Philadelphia chromosome that describes all of this. It was essentially, all of these discoveries were made in the medical hospital in Philadelphia, and so this chromosome has been named as the Philadelphia chromosome. Once one has understood that this is the fusion protein that's causing this leukemia, one was able to understand the various kinds of domains of this protein and be able to block this protein by getting hold of a molecule. And so now there is this new drug called, not new anymore, but there's this drug called imatinib, which essentially blocks the impact of that fusion protein, as a result of which lives of children are saved. So one way to save the lives of children was to be able to understand how that leukemia happens, and the fusion protein was identified. A lot of structural biology work on the fusion protein was done. A molecule was developed that binds to this particular fusion protein, as a result of which the negative impact of the fusion protein does not happen in the child, and therefore the child is saved. So that's, again, genomics, understanding of genomics, research in genomics is saving lives of children. I'm going to spend the last few minutes of my talk describing our own work, but again, saying a few more things. I'm going to skip a few of these. Okay, saving lives of men is what I want to talk about. So, I've talked about saving lives of women using genomics. I've talked about saving lives of children using genomics. Now I want to talk about saving lives of men using genomics. And I will begin with 2 stories. Both of these 2 stories are correct, are factual. My story number 1 comes from the pages of the New England Journal of Medicine, which is the oldest medical journal in the world. So this is the story. The New England Journal of Medicine produced this picture in one of its pages, and this is, of course, a patient with cancer, and all of these, each dot is a tumor, so you're covered with tumors. This person came to the doctor, and the doctor prescribed a medication, and after about 3 weeks at a particular dosage of that medication, what had happened to this individual is that he underwent another CT scan, and essentially what one sees is that all the tumors from the body have evaporated. So this is good news. All the tumors have evaporated. So essentially, the patient has been completely cured, at least temporarily. It might come back, but at least temporarily has been cured of tumor. The same, the doctor encountered another patient with essentially the same kind of features, and I'm not reproducing that picture. By and large, it's the same kind of a picture that was produced in that journal. And the doctor again treated this patient in exactly the same way, but you can see what happened to this patient after 3 months. 3 weeks, nothing has happened to this individual. All the tumors remain where they were before the treatment. So the question that we want to ask is, why is there a smiley here and a sad face here? What makes this treatment, what makes this individual respond to the treatment so well, and the other individual who comes with the same kind of features does not respond at all? So that's essentially the research that needs to be done. If you look at a cancer genome, cancer usually involves multiple mutations. So, like I said, that most mutations are not even inherited. Most of these changes are not even inherited. So they happen during the life course of that particular individual and will remain with that individual until the death of that individual. And more changes will take place in the cells. So imagine, if you will, that this is the epithelial layer, and these are all epithelial cells below the epithelial surface. So these are all cells. These are cells that we are born with. Every cell looks exactly the same, but during the life course, some cells acquire new mutations. So new mutations arise in some cells. Most of these mutations will be innocuous. We would not even know the existence of these mutations. Of course, then again, on some new mutations will appear. Some of them on the background of the old mutations, but some of them on new cells. The mutations will appear in new cells as well. And some of these mutations might actually give the cell a growth advantage, in the sense that the cell will make multiple copies of itself become a clone. But the clones are very small, and these clones are happening in us all the time, and we don't even know the existence of these clones. So here is another mutation that has arisen, and it gave rise to a new clone. And on the background of all of this, mutations continue to happen. So here is a mutation that took place just now on the background of 2 previous mutations. But this mutation we expect would be as innocuous as the previous mutation, but it was not. This mutation resulted in a huge proliferation of cells. It breached the epithelial layer and became a frank cancer. So it became a solid tumor, and the final mutation that drove this to becoming a solid tumor was this red mutation. So this red mutation is a driving mutation for this particular kind of tumor. So what we like to know is, in which particular gene did this mutation take place? In other words, for any kind of cancer, what we really like to know, because somatic mutations are happening, what we would like to know is, where are the drivers, what are the driver genes, and what are the driver mutations that causes the normal tissue to becoming a tumor tissue. So that's essentially what, and even after that red mutation has happened, there are multiple mutations, that there are no new mutations that are happening. Again, most of these mutations mean nothing to us, but this red mutation meant a lot to us. So the question is, what are the genes and genomic alterations that drive? And drive here means that the cells get a growth advantage, and they proliferate, and they clonally propagate and become a solid tumor. So that's the driver mutation that we talk about. Like I said, that I've spent the last 10 or 12 years researching on cancer of the oral cavity in India. And let me begin by telling you a little bit about the epidemiological features of this kind of cancer. It's the 8th most common cancer in the world. About 260,000 new cases appear annually. 2/3 of them appear in developing countries, and it accounts for 1/3 of all tobacco-related cancers in India. In some places, one finds that between 20 and 60% of cancers of the oropharynx, of the oral cavity and the pharynx, is associated with the human papillomavirus. The human papillomavirus is the causative virus that causes cervical cancer in women, but it's also sometimes, in some countries, it's associated with oral cancer as well. In India, we did not find it to be associated with oral cancer. Human papillomavirus is not associated with cancer of the oral cavity in India. We carried out, so this is data from about 200 patients, and from each patient we collect paired blood sample. We collect the blood sample and a tumor DNA and a tumor tissue, and we isolate DNA from both. The blood sample carries information on what's been inherited. The tumor tissue contains information on changes that have been inherited, plus the changes that have been acquired during the lifetime of this individual. So the blood contains germline information on germline mutations. The tumor DNA will contain information on both germline, as also somatic mutations. So if we take the repertoire of mutations in the tumor DNA and subtract out, so to say, the repertoire of mutations in the blood DNA, we will be left with somatic mutations only. And then we can analyze the somatic mutations and ask ourselves, what are the driver genes that drove through somatic mutations for a normal tissue to become a tumor tissue? So what we've done is we've now expanded. So this is n equal to 200. We now have reached the stage of n equal to 500. We haven't reanalyzed our data in full, but when we had reached about 450, we had reanalyzed our data, and we don't find any major changes between what we had, our inferences from n equal to 200 patients to about 450 patients. Essentially, there are these genes, some of which may be familiar to you. P53 is a gene that's involved in many kinds of cancer. Caspase 8 FAT1. So we have a bunch of about 10 genes that drives the normal tissue to mutations in which drive the normal tissue to becoming a tumor tissue, and TP53 tops them all. So what we find is that of all of these driver genes, most of these genes are behaving as tumor suppressors. Just to recall, tumor suppressors are those genes that are normally expressing all the time, and eventually mutations in those genes will obliterate the production of that normal protein, as a result of which the control on cellular growth will be obliterated, and therefore this particular gene is then this particular protein is then going to not be able to control, and therefore cancer will appear because of uncontrolled cellular growth. What we find is that tumor suppressors drive oral cancer of the oral cavity, and that's bad news, because that's like a normal protein that's expressing all the time, and mutations are killing that particular protein. So you can't reverse the mutation and get the normal protein back, and therefore cancer being driven by tumor suppressors is always a bad news, because it becomes very difficult to manage such cancers. And oral cancer in India, oral cancer in general, not just in India, oral cancer is driven by tumor suppressors, and therefore oral cancers are going to be very hard to control epidemiologically. But what one could do, the good news is that one can control the use of tobacco, especially chewing tobacco, in which case you would not have the carcinogens hitting the cells and causing those somatic mutations. So once the somatic mutations are caused, it's bad news because you can't control it. But you have these environmental factors that you can control that will prevent the causation of these somatic mutations. So one needs to strike at the root, and the root is this environmental factor called tobacco, and the way that tobacco is used by individuals who have cancers in the oral cavities to chew, not smoke. When you smoke, you get more of a cancer of the pharynx and of the tongue, as opposed to the epithelial squamous cell carcinoma of the oral cavity. What we did then was to look at these patients who have these kinds of somatic mutations and the driver genes. I won't get into the gory details of it, but essentially what we did was to look at these mutations and mutational profiles of the patients, and we were able to derive molecular subclusters of these patients. So we derived 3 molecular subclusters of these patients, as you can see, and each of these molecular subclusters had their own repertoire of mutations, of driver genes and mutations, and they had their own profiles of survival. Some members of one of these clusters, or two of these clusters, had very high survival probability, the others did not, and therefore molecular profiling actually helps us to understand which genes lead to earlier death of these patients who because of those mutations in those genes get tumors. What we were also able to do is to be able to map these mutations on pathways. And what we found was that the arachidonic acid metabolism pathway that regulates inflammation and tumor progression actually is enriched by these various genes and mutations in these genes. And there are these various kinds of molecules that can block arachidonic acid metabolism pathway, and these molecules are called COX and LOX, and what they do is the changes in COX and COX loss will enhance proliferation, enhance anti-apoptosis, and so on and so forth. So these are molecules that can be effectively used to manage or to treat oral cancer. And as a matter of fact, if you do treat oral cancer using COX and LOX molecules, you would find that those individuals would survive better. The survival probability after the enhancement of survival probability is about 17 months. And LOX and COX, by the way, are very cheap. Aspirin is molecule that belongs to this class. And so just treatment with aspirin will enhance the survival probability by 17 months. 17 months may not look like a big time, may not look like a long time, but 17 months is for a family that has a cancer patient. 17 months of additional survival is good. 17 months is a long period of time. And so, and just treatment with a very cheap medication such as aspirin does the job. So that's a conclusion that we got after doing all of these molecular analysis of these 500 patients and identifying a particular pathway that enhances survival or that reduces survival, and blocking that particular pathway with a medication as cheap as aspirin will enhance the survival probability by about 17 months. So we published these by and by. So I come back to my story, the story that I began with, and the question that we asked is, why is there a smiley here and a sad face here? So the answer to that question is what I will provide now. If you look at a particular gene called BRAF, these mutations in BRAF are found in malignant melanoma and other cancers. They're found in 70% mutations in BRAF are found in 70% of malignant melanoma, 30% of thyroid cancer, 10% of colorectal cancer. So BRAF is an important gene, and mutations in this gene will cause cancers of various organs. And what does this BRAF gene do? BRAF gene, there's a lot of trafficking that takes place from outside of the cell to inside of the cell, and these are various kinds of signals that come from outside of the cell, telling the cell what to do and what not to do. BRAF is one such molecule, BRAF is one such protein that regulates the amount of cargo that comes from outside of the cell to inside of the cell. And anytime there is a mutation in BRAF, that regulation of the cargo from outside to inside is hampered, and certain things happen because of that dysregulation of the cargo. So that's what this cartoon shows, that there is this cargo that happens, and BRAF essentially signals the cargo. Well, these entities that are coming from outside of the cell is telling the cell to grow, but the BRAF also knows when to stop production of its own protein, such that the signaling won't happen and the cellular growth will stop. So at some point of time, the cellular growth needs to stop. And if it doesn't stop, then of course you know what's going to happen. Tumor is going to arise. And so it's involved in sending signals inside cells, which are involved in directing cell growth. So when there is a mutation in BRAF, it does not direct cell growth in the way that it should. And as a result of which, when you have a mutated BRAF, this signaling process is gone, it goes berserk, and as a result of which the cell grows and grows and grows, and that's the hallmark of any kind of cancer. Cellular growth is the hallmark of any kind of cancer. So this is what one understood. Then what one needs to do is, in order to stop this process, you need to identify a drug that will act against the BRAF, the mutated BRAF. But you have to be careful that the drug should not act against the normal BRAF, because if you inhibit the normal BRAF, then you are going to obliterate its function, as a result of which you are going to artificially stimulate formation of tumors. So one needs to be careful, one needs to get a drug that will only act against the mutated BRAF, and as a matter of fact, such a drug has been found. Actually, the BRAF mutation BRAF gene has multiple mutations, but fortunately there is one common mutation that's called D600E, that's in the amino acid position 600 of that particular protein. It behaves as an oncogene, and a drug has been found that will act against only this particular mutation in the 600 amino acid position, and that drug is called Nayudamma, which is a blockbuster drug. It inhibits the BRAF if this particular mutation is present, and this has become a blockbuster drug. As a result of which, you get a face that is smiley and a face that's sad. And so essentially what happened is that this particular patient, the first patient, was treated with vemurafenib because the patient had a BRAF mutation and this V600E BRAF mutation, as a result of which the patient responded so well, because that acted against that particular mutated BRAF, did not allow the BRAF to send wrong kinds of signals, and therefore the cellular growth was stopped. On the other hand, the second patient, unfortunately, did not have mutations in BRAF, yet vemurafenib was prescribed, and vemurafenib, of course, will not act against a non-mutated BRAF because it was meant to work only against V600E mutation. So it actually did not work. This particular patient had cancer, but that was not related to BRAF, was prescribed BRAF by the doctor because the doctor had such a great success with respect to the first patient. The doctor did not imagine that the second patient did not have BRAF mutation, prescribed the same drug, and ultimately failed. The patient failed to respond, and that was a sad story. And so you can imagine that how precise treatment has become. You look at a particular mutation in a specific gene, there is a molecule that will act against that particular mutation and will save lives of people. So that's what it is. So it's actually valine to glutamic acid. So that's the reason why it's called V600E. And the reason and the way that it has this drug has been named is VENURAFNEID. So vemurafenib is what the drug name is. It comes from the valine to glutamic acid mutation in BRAF. That's how the drug has been named. So I will sort of end with this particular slide, and I've told you how genomics, understanding of the genetic basis of a disease, especially of cancers, are saving lives of women. And I've given you examples of women surviving from breast cancer. I've given you examples of children surviving from leukemia. I've given you examples of men surviving oral cancer because of these various kinds of research that has gone on in the last 2 decades. When we have the technologies to be able to understand the somatic basis of these kinds of mutations or these kinds of changes. So the paradigm of medicine has actually shifted. It's shifting more and more, but it has shifted in countries where this is practiced now. In the older regime, essentially medicine was very descriptive. Now one needs to understand the mechanisms of disease. One's understanding the mechanisms of disease. Earlier, you tried and failed. Sometimes it succeeded, and those were empirical diagnoses. But now it's all mechanism-based diagnosis of treatment. You try and understand what that protein does, and then you try to block that particular protein if it is sending wrong kinds of signals and so on. It's grouped by organ or site. I've given you the example of BRAF. BRAF is involved in colorectal cancer, melanoma, and multiple kinds of things. So it's also subgrouped by molecular or biological classification, just as I've shown you for the BRAF V600E mutation. If an individual had, or a group of individuals had similar looking symptoms, clinical symptoms, they would be treated uniformly. But today, because of the advent of genomics, we are getting individualized treatment because we are first getting ourselves treated, getting ourselves diagnosed using what kind of changes we have. If we genetic changes we have, if we have understood the genetic changes that underlies that disease, and then we get these individualized treatment. We retrospectively diagnose disease. Today, disease is evaluated prospectively. We try and do genome sequencing to understand prospectively what particular disease that this individual may have, what subtype of the disease this individual may have, and then using that information on the subtype appropriately provide molecules for treatment. Earlier, only when the patient became acutely ill, we took care of the patient. Today, because of this prospective nature of identifying, prospective nature of diagnosis, we can detect individuals early, sometimes even before clinical manifestations of the disease, intervene using appropriate molecules and be able to contain the disease even before the disease has onset. So this is essentially what genomic medicine is all about, understanding the genetic basis of a disease, using that information in order to design molecules and identifying individuals early with respect to what kind of genetic changes they have, and then being able to manage the disease in a individualized or more precise manner. That's what genomic medicine is all about, and that's the reason why there is so much of excitement about genomic medicine, and it is essentially free of gender, it is essentially free of age. It's impacting on lives of every individual in the society. And there's going to be more and more of these kinds of individualist medicine, and that's the reason why those of us who are in this game are very excited, and because it's impacting on lives of women, children, and men. Thank you very much.
Great informative insights about the role of genomic medicine on augmenting the survival rates of population with cancer, be it breast cancer in women, leukemia in kids, or oral cancer in men, or any other type of cancer. Thank you so much, sir. May I now request Dr. K.J. Sriram, Director, CSIR-CLRI, to present the memento to Professor Partha P. Majumder. Thank you, sir. I would like to thank one and all present here for your gracious presence. My heartfelt thanks to Professor Partha P. Majumder for delivering the Nayudamma Lecture 2023. I thank the former directors, members of the industry, members of leather associations, CSIR family, students, and all other stakeholders for your unstinted support to the Institute in all our endeavors. I thank the media personnel. I thank all the members of the organizing team and the support team, engineering services, information technology, AC and instrumentation, and lab maintenance for their unstinted support. Finally, I thank our director for his immense support in organizing this event. I thank you all once again for your gracious presence in the Dr. Nayudamma Lecture 2023 and making this event more memorable. Thank you. Thank you so much. May I now request you all to please rise for the national anthem. Thank you, one and all.